Good morning and welcome to your seed dissection. You have no idea how many times I've recorded this, so hopefully this one will be successful. What you need is a bean, or you can just watch me and draw along with my pictures as you go. You need a bean that you've preferably soaked for up to 12 hours. If you haven't quite managed to do it for that long, have a go, see if it works anyway. And you can always do this at a later date. So I've soaked my bean in water. Overnight I have. And what it's done, just by looking at the comparison between what it looked like before and what it looks like now, is it's really swollen up. Although the seed was really firm, it's quite swollen with water now. It's lost the wrinkles that were on the outside and it looks a lot plumper. First thing in a dissection that we should do is we should draw what we're starting with. So if I look around the outside, I've got this tester. Sorry, my hands are wet from all the water. The tester on the outside, which is the seed coat. Now remember, this was the ovule. Before fertilization took place, this was the ovule. And around the outside then, that seed coat. And we've got a little hole at the bottom here. And this is called the micropyle. Now this is different size in different seeds. Beans are a lovely seed because they're so big, so very easy for us to dissect and to have a look at. So they also germinate really quickly. You could do this with other seeds, but it's a lot harder to see. Now, what you need to do is once you've drawn this and labelled the outside, so we've got the micropyle at the bottom, that's where the pollen tube would have entered and fertilised with the pollen nucleus coming down, and not a lot else. So what you need to then do is very carefully try to peel away the seed coat. Now, this is my second seed of doing this, and the seed coat doesn't come away as easily as the previous one. I've just made a tiny little cut into the back of it so that I can peel it away. And it's not fully attached to the inside, so it does come away quite easily. You've just got to give it a go. Now, I'm sure some of you are going to laugh at this because the inside of the seed does look quite funny. Have a look at the one you've got. I'd like to see your labelled pictures of these. So here is the inside of my seed. The seed will very, very easily split in two now, so it doesn't matter if it goes into two. And what we can see are two significant structures. You see this pointing down one? This is going to eventually become the roots of the seed, and it's called the radicle roots, RR. That's the embryonic root. We've then got this section just above, if you can see that there. It's probably clearer to see on the diagram on page 21. And that is going to become the shoot, and we call that the plumule, the embryonic shoot. Now, you can take your seed apart, obviously drawing at each stage. Well, this one's much nicer than my last one. And right in the centre, I know the picture quality isn't the best on here, but bear with me. Hopefully you can see what I'm doing if you're doing it with me. What you can see there is the embryonic shoot, the plumule, and the radicle, the embryonic root, pointing down towards gravity. There we go. Now we've also got to label this centre part. This whole of this centre part here, this lighter yellow, you can see, is called the cotyledons. The cotyledons are sometimes called the first leaves. And the reason they're called the first leaves is because they provide all of the energy that the plant needs to grow until the first leaves can take over and begin photosynthesizing. So in order to do this, the cotyledons are a massive starch store. The cotyledons are a starch store, and when the water enters the seed, it begins to break down that starch back into glucose. So remember, we've talked about starch as being chains and chains of those hexagon-shaped glucose molecules. All that's needed to break that back down is water. Water breaks down those bonds and it ends back up as glucose. And that glucose can then be used. We can't photosynthesize, but to make energy or to generate energy, this seed now has to respire. Remember respiration? Glucose plus oxygen goes to carbon dioxide and water. So we need glucose that glucose is in the seeds stored as starch. So as soon as we've added that water, that starch has become activated into glucose, so we can use it. Oxygen. Definitely need oxygen for respiration to take place. How is it going to get there? Well, the seed in the first place, when you add water, swells. The water enters through the micropyle, which causes the whole of the seed to swell up, and it cracks the outside of the tester, the coat. That cracking causes more holes so that water can enter through those, and also oxygen can enter. So we're adding water to break down the starch and oxygen just through the gaps in the seed coat. This is why it's really important not to flood seeds with loads and loads of water. Some seeds there are exceptions to, and you need to do this. But most seeds just need a small amount of water in order to split open that coat to get the oxygen in. Once that oxygen is in, it can react with the glucose to release energy. Now that energy, respiration, is going to be used to grow that radicle and the plumule into the shoots and the roots until the first leaves are formed and we get some greenery on the plant, and at that point photosynthesis will take over. So it's all stored in there. All the starch, all the requirements for growth that is needed in a seed is inside this bean. Now, if you don't have a bean and you'd like to try this with any seeds that you can either try it with a sunflower seed, why not? And what's really important is that you soak it first. The longer you soak it, the easier it is to split, and it will begin to germinate. If you wanted to leave it longer and you could actually germinate the seed before you then dissect it, that would be really interesting too. I really look forward to seeing your pictures, your annotated diagrams, and if you're brave enough, why don't you do a video like I've done? Enjoy!